find ways, to divide abnormally. And these hallmarks are explaining, how cancer normal cells turn into malignant cells. By sustaining proliferative signaling, they evade the growth suppressors, they resist the cell death. And these hallmarks from 6 and are now gaining numbers to 14. So we know that since cancer is so complex, so one size treatment fitting to all the types of cancer won't work. But when we talk about cancer and how we can treat them, we can see that with the same type of cancer, having same cancer cells, but some patient may find the target and some may not find the target. So the conventional treatment usually are surgery, chemotherapy, radiation therapy. But now one approach to treating cancer is precision medicine, where we use molecular profiling to personalize cancer treatment. Of course, cancer treatment is personalized in a number of ways. Apart from molecular profiling, the patient's age, the type of cancer, and many other factors are taken by the oncologist to determine which type of treatment will be the best for the patient. Now, when we talk about biomarker guided therapies in precision oncology, it is seen in many studies that they have led to better outcomes than the traditional methods. Because when we talk about the conventional treatment like the chemotherapy, they attack all the cells of the body, be it the cancer cells or the healthy cells, and those come with many side effects. But targeted therapy are designed in such a way that will mostly attack the cancer cells with less toxicity and are shown to be more effective. So those hallmarks of cancer that has been studied so far, they provide a framework to support the designing of targeted drugs for the cancers. Now, when we talk about the classes of targeted cancer therapies, so they could be of two types mainly. We can talk about small molecule drugs. So those are, you can say, the size less than 900 daltons, and they penetrate cells and modulate cell signaling pathways. Then we can talk about monoclonal antibodies. Those are larger in size, larger proteins, more than 900 dalton, and they target the extracellular proteins on the cancer cell. And when we talk about antibody drug conjugates, so they combine the monoclonal antibody with cytotoxic agents so that the drug is delivered directly to cancer cell. And this has also shown to be improving clinical outcomes a lot. And let's now move to what are molecular biomarkers. So when we talk about molecular cancer biomarkers, they are any measurable molecular indicator of risk of cancer, occurrence of cancer, or patient outcome. So biomarkers may be divided based on the genetic makeup of the patient. For example, germline or somatic variants. Germline variants are inherited variants present in all the cells of the body. Somatic variants are acquired variants that are present in the tumor cells. Again, epigenetic markers. Those reflect DNA methylation or histone modification patterns regulating gene activity. For example, MGMT promoter methylation for glioblastoma. So those will predict if chemotherapy, alkylating chemotherapy would be responsive. Then we have the new evolving signatures of biomarkers, the transcriptomic signatures, which are the RNA expression profiles, and the proteomic signatures, which mostly by IAC detects differences in protein expression. So what are the clinical impacts? Is when we talk about prognostic biomarkers. So those are related to the disease outcome. And then we can talk about predictive biomarkers, which are useful for evaluating drug response or therapeutic intervention. For example, we'll mostly talk about the therapeutic intervention predictive biomarkers today. And yes, we have pharmacodynamics, which will see which drug is going to work for which patient. Yeah. So with a vast majority of biomarkers detected, what are the technologies that we can use? We can use next generation sequencing technology, which is a massive parallel sequencing of so many thousands of genes, and we can detect various genetic variants like single nucleotide variants, like small insertion or deletions which are less than 50 base pairs. We can detect copy number amplifications, we can detect fusions, and then we can detect genome-wide biomarkers, which are HRD, MSI, TMB, which we'll be discussing about in the later part. And next, we can detect by PCR. What's PCR? It is a polymerase chain reaction, and mostly used to detect known and specific mutations. Then comes MLPA, which is multiplex ligation dependent probe amplification, and this technique is used to detect large genomic deletions or duplications. And yes, the standard ISC immunohistochemistry, which will detect protein expression on tissue, for example, PD-L1 expression, which guides immunotherapy response. Yes, and this is a study where there was a biomarker driven study to see how much percentage of patients are eligible for a biomarker informed therapy, whether it be approved therapy or whether it is on late stage clinical trials. So it is seen that in a wide variety of cancers, almost 30% of patients are eligible for biomarker informed therapies. Is my slide moving?
No,
So,
the screen share was stopped actually.
okay, one second. Is it showing now?
Yeah, now it's visible.
Okay. Yeah. There are some technological problems here. So when we talk about, there are a few examples of key actionable targets in some of the common cancer types. For example, EGFR, ALK, KRAS, ROS1 mutation in lung, BRCA1 and BRCA2 mutations in breast, KRAS, NRAS, BRAF mutations in colorectal cancers, KIT mutations in gastrointestinal cancers. So these are a vast, data of we have so many biomarkers for so many types of cancers, of course, but guideline based therapies, for detailed information we can always look into the NCCN guidelines to see what kind of drugs and which gene has been involved, so that we can see if there are any FDA approved drugs, if there are any clinical trials. So all this information will definitely help the patient and the clinicians to decide the right treatment. So these are the when we talk about somatic variant classification in cancer. So this is AMP ESCO CAP guidelines, where we have tier 1 variants, which are variants of strong clinical significance, tier 2 variants, which are variants of potential clinical significance, tier 3 are mostly VUS, and tier 4 are benign. Again, we have different evidences which support the therapeutic prognostic and diagnostic evidence. For example, when we talk about level A evidence, we have FDA approved therapy and which are included in professional guidelines. When we talk about level B, they have well powered studies with consensus from experts in the field. Again, we have level C evidences, which could be FDA-approved therapies in other tumor types rather than the tumor of the patient. And level D evidence would be preclinical trials or a few case reports. So this is how we go about the classification of somatic variants. Next, we'll go on to looking into a bit about the immuno-oncology markers, which are MSI and TMB, and PDL1, it's a ISC marker, and which we've already spoken about. So when we talk about MSI, so what is microsatellites first? So they are DNA sequences of 1 to 6 nucleotide base pairs, and it is distributed mainly in the non-coding regions of the genome. And it could be, in the telomeres, in the intronic regions, in the promoter regions. Now, what is the function of the microsatellites in the human nuclear genome? So this is involved in chromatin organization, cellular senescence, gene activity, DNA repair, and many other functions. Now, when we talk about microsatellite instability, so this is a condition of genetic hypermutability which is caused by DNA mismatch repair, MMR dysfunction. Why do we test MSI? Because that can help guide immunotherapy treatment like PD-1 inhibitors. So when we talk about microsatellite instability, this could be due to sporadic reason or due to inherited conditions like Lynch syndrome. So sporadically, you can see MSI high, and we can see mostly in colorectal cancers which are BRAF B600 positive or which have MLH1 hypermethylation. So those could be sporadic, and when we have a germline mutation in any of the MMR genes, so this could be associated with Lynch syndrome. MSI can be detected by different methods. First, first we can detect MSI by ISC, so which detects the loss of expression of key MMR proteins, which are MLH1, MSH2, MSH6, PMS2, and loss of staining for one or more of the MMR proteins. They indicate deficient MMR. So here antibodies are used to stain the tumor tissue. Again, MSI can be detected by PCR on NGS2. When we detect MSI by PCR, so it detects instability by comparing the lengths of microsatellite markers in both the tumor DNA as well as the normal DNA, which is the blood. Now, when the MSI is unstable, when 2 out of the 5 markers are unstable, and they're in but this is the guidelines. We have 2 mononucleotide repeats and 3 dinucleotide repeats. Next, MSI by NGS is a is a widely used technique nowadays because it is combined with the comprehensive genomic profiling, and it allows high throughput sequencing of numerous microsatellites and genes, and it enables simultaneous determination of both tumor mutation burden too. Now, identifying MSI or MMR deficient tumors is critical for treatment selection for immunotherapy, particularly in cancers like colorectal cancers or endometrial cancers or biliary tract cancers and so on. Yes, FDA has approved MSI as a tumor agnostic marker. That means this is approved for immunotherapy MS as a marker regardless of the tumor type. Yeah. So this is a study of mismatch repair deficiency across 12,000 tumors by NGS, and it was found that MSI mismatch repair deficient tumors were identified in 24 out of 32 tumor subtypes tested, uh, more often in early stage, and this is a good number. So this MSI high offers distinct data, as I have already spoken, regarding immune checkpoint inhibitors, in addition to the data available from TMB and PDL1. Next, going to tumor mutation burden. So tumor mutation burden is a number of mutations mutations per megabase of coding DNA within a tumor sample. And it is also a predictive biomarker for benefit from immune checkpoint inhibitors. Cutoff uh, is 10 mutation per MB, which has been approved from different trials. And FDA has also approved it as a tumor agnostic marker for patients with advanced stage solid tumors following disease progression on standard of care therapy who lack an alternative treatment option. So different parameters influence TMB. For example, the size of the panel, the sequencing strategy. So these has to be taken care. And again, though it has been approved as a tumor agnostic marker, but it is different for different. The response would be different for different cancer, which the clinician takes into account before deciding the therapy. Yes. Now, how the immunotherapy works. So what happens is T cells, can recognize and kill abnormal cells, right? But the tumors develop the mechanism to escape this. And when PD-L1 on the tumor cells binds to PD-1 on the T cells, and this immune activity is lost. And blocking this interaction with immune checkpoint inhibitors restores the T cell function and enables the T cells to attack the cancer cells. Yes, so these are some of the tumor agnostic biomarkers like MSI high or deficient MMR, TMB high solid tumors, and for immune checkpoint inhibitors, BRAF V600 for BRAF V600 mutation, BRAF inhibitors, then antibody conjugate drug for HER2 positive solid tumors, trastuzumab. Then we have anti-RK inhibitors for anti-RK fusion positive solid tumors, RET inhibitors for RET fusion positive solid tumors. Yeah. And yes, moving to another biomarker is homologous recombination repair deficiency. Now, what is homologous recombination repair? So it is a high fidelity process by which cells repair DNA double-strand breaks and is very critical for maintaining genomic stability. The genes which are involved in the HRR pathway, if mutated, it could be tumor-specific or somatic mutations or germline mutations, they cause HRR impairment, right? So in the image, we can see that uh, there is single strand break, right? There is this PARP enzyme which helps to repair single strand break by base excision repair, and the cell survives. Now, homologous recombination deficiency has been shown to be associated with sensitivity to platinum-based treatments and PARP inhibitors. How these PARP inhibitors work? So the PARP inhibitors uh, lowers the power of the PARP enzyme for single strand break. And this leads to replication for collapse, causing double-stranded breaks also. In cells which has homologous repair propagation, they will repair the double-strand breaks and the cell will survive. But when there is a homologous repair deficiency, the cells will be unable to repair the double-strand breaks and then leads to cell death. So this is the concept of synthetic lethality, in which two conditions, that is PARP inhibition and HRD, will lead to cell death when combined. So impairing both the single-stranded breaks and double-stranded breaks. Uh, Now when we talk about genome-wide HRD scoring, so HRD could be you know, measured by different genomic scars. All right. When we talk about single measures, that could sometimes be insufficient. So HRR mutations are one way where we can see if there is a change in the HRR mutations. Those tumors may respond well to PARP inhibitors. But apart from that, there are some genomic scars which are loss of heterozygosity, which are telomeric allelic imbalance, which are large scale transition. So these reflect the level of homologous recombination deficiency across the entire genome. So there are scores which will measure and provide a comprehensive metrics to see if the tumor is HRD positive or HRD negative. So when we talk about the clinical impact, HRD guides PARP inhibitor use across multiple tumor types such as ovarian cancer, breast cancer, pancreatic cancer, and prostate cancers. Yes. Moving ahead, excuse me. Moving ahead, let's talk a bit about liquid biopsy. So this it has come as a rescue, liquid biopsy, when when tumor tissue is unavailable, or the biopsy of the tissue is not feasible for the patient at the current time. So this has come as a rescue, which is why it is widely being used these days. So it's a minimally invasive technique that analyzes tumor-derived materials such as ctDNA, CTCs, exosome from bodily fluids like blood, CSF, for cancer diagnosis, treatment guidance, and real-time monitoring of disease progression or relapse. When we look into liquid biopsy, there is an evolving biomarker, which is a CTC, circulating tumor cells, and those are the cells that are shed into the bloodstream by the primary tumor. And they have a diagnostic, prognostic, and predictive they can be used as a diagnostic, prognostic, and predictive biomarker in many types of cancers. And studies are being growing, regarding the findings of CTC and how it could relate to metastasis. But yes, the approvals or in the guideline-based testing for CTC is still yet to come. When we talk about ctDNA, again, is cell-free DNA fragment in the bloodstream that originates from the malignant tumors or the circulating tumor cells. These ctDNA carries genomic alterations specific to the tumor, and its analysis will allow for a detection of tumor-specific alterations. Yeah, and you can see in the diagram here that cell-free DNA, which is in blue, and ctDNA, which is in green. So when we talk about cell-free DNA, these are small DNA fragments in circulation, and they are released in bloodstream due to cell death, and mainly from hematopoietic cells. And circulating tumor DNA, these are small fragments that are released in circulation from cancer. So there could be 1 to 5 mutant tumor DNA fragments in, say, 10,000 cell DNA fragments. So detecting ctDNA is like a needle in a haystack and requires sensitive test methodologies. And these are the recommendations of ESMO regarding the use of circulating tumor DNA assays for patients with cancer. And we could have potential clinical applications in if after definitive local treatment for molecular residual disease detection, to see if there is a molecular relapse in early stage cancer after local treatment. Then for advanced diseases, to see if there could be biomarkers to guide therapy decisions, or to see if there could be adjuvant therapies which could be escalated or de-escalated based on the ctDNA positivity or negativity. Then we can use it for monitoring response and also for identifying if there are any resistance mutations in that has been developed. So let us take an example of ctDNA, how it has been used currently in non-small cell lung cancer. So it can be used for first and second line therapy selection. For example, if an EGFR mutation is detected in ctDNA, it could guide treatment selection. For example, EGFR tyrosine kinase inhibitors. Again, after surgery, if there is no ctDNA detected, clinicians can take decisions on if we need a treatment de-escalation. All right, and if there is a ctDNA, for example, residual disease detection, sometimes when the patient is on a particular TKI, and then we can also, as time grows, the cancer cells may develop resistant mutation. For example, EGFR T790M. So this mutation is a resistance marker for first and second generation TKIs. So in that case, third generation TKI or similarly will be more responsive for the particular patient. So yes, this has been some of the utility. And again, there are several trials. For example, the dynamic trial in stage 2 colon cancer, and it was in 455 patients who underwent randomization. 302 were assigned to ctDNA guided management and 153 to standard management. Now, ctDNA guided approach reduced the number of patients who received adjuvant therapy, and yeah, the risk of recurrence was not altered. And again, ctDNA positive patients appeared to derive considerable benefit from adjuvant chemotherapy treatment. So there was very low risk of recurrence in untreated ctDNA negative patients too. So this you can see the recurrence-free survival, 96.6% in the standard management. Uh, 90, sorry, in a follow-up of 24 months, it's 2 years, you can see that standard management, we can see 93.5, and ctDNA guided management 92.4, just a difference of 1.1%. So this ctDNA guided approach to the treatment of stage 2 colon cancer reduced adjuvant chemotherapy use without really compromising the recurrence-free survival in those colon cancer patients. Yeah. So now let's discuss some of the few case studies. So we'll go with the case of adenocarcinoma colon. So this is a 92-year-old male, and who has a history of carcinoma prostate in 2007, and was diagnosed with colon cancer in 2024. He has completed 6 cycles of chemotherapy, 7 cycles of immunotherapy, and in December 2024, and but PET-CT in 2025 suggested of progressive disease. A molecular testing was done in yeah, in tissue, blood, and pleural fluid sample, and KRAS G12C mutation was found, which is a tier 1 level A pathogenic variant. When we talk about the other variants, there were ATM mutations detected. One is a CHIP mutation, one is a true somatin mutation, and other level 2 genetic variants were found in TGF beta, TGF BR2, GNAS, and MEF2D. This KRAS G12C variant, uh, when we talk about it, the alterations in this KRAS gene instigate the downstream pathways like RAS, MAT, ARC, and thereby it fosters carcinogenesis and tumorigenesis. Now, according to the NCCN guidelines, we do have KRAS G12C inhibitor, sotorasib or adagrasib, plus anti-EGFR TKI therapy for approved for these patients. And yeah, so when we talk about other variants here, ATM is associated with HRR gene pathway aberration, but approval for colon cancer that has to be, of course, not a tier 1 variant. Again, CHIP mutations. So those are the mutations which are somatic mutations derived from hematopoietic cells, but they are not from tumor origin. So that has to be that differentiation has to be very well taken. Yes, the FDA granted accelerated approval for aggressive cetuximab for KRAS G12C mutated colorectal cancer. And this could help the patient to have treatment options for better response. Yeah. So next case is a case of ovarian cancer, 53-year-old female. So previous test by IST which showed microsatellite stable, and by NGS it was detected as MSI, and so the patient was treated with immune checkpoint inhibitor, but there was progression again. Now, comprehensive testing was done on both tissue and plasma sample. Same, we can see the TP53 variant in the ERCC2 variant, which was detected earlier in the tissue to be present here, and there is a TMB which is high. Again, but when we see on the ctDNA or the ctDNA or detected by the blood sample, we could see an additional variant which is the BRCA2 variant. And apart from that, the TP53 ERCTC2 variant is there. Again, we see a variant in STK11. So STK11 is a known marker for resistance to immunotherapy. So the patient has been treated with immunotherapy for so many months, and but still was not responsive. So this could be said as a financial toxicity also, because immunotherapy is quite expensive. And when we talk about time, yes, for cancer patients and taking treatment decisions, time is a very significant factor. Again, we here know that the immunotherapy might not work well for this patient. And since there is a BRCA2 variant, we have options for PARP inhibitors or a combination therapy which the clinician could look into. Next case is of adenocarcinoma rectum. This is a 27-year-old male, and he has been diagnosed with moderately differentiated adenocarcinoma of rectum, and HER2/neu was positive, and he has received 2 doses of trastuzumab and 4 cycles of chemo, and it is suggestive of disease regression. So yes, we know that there was already HER2/neu positive, and which is ERBB2 gene, and this was a molecular testing in the blood which shows ERBB2 amplification, 11 copies. And what are the clinical implications? So ERBB2 or HER2 is an EGFR receptor tyrosine kinase, which activates the PI3K, KT, mTOR, and the downstream pathway, and thereby promoting tumorigenesis. Anti-HER2 therapy with signal transduction inhibition, like trastuzumab, is indicated in HER2-amplified tumors that are also RAS, BRAF, wild type. And moreover, we have trastuzumab deruxtecan, which is antibody drug conjugate, which is also indicated in HER2-amplified tumors which have ISC positive. Yes, this is FDA grants breakthrough designations to trastuzumab deruxtecan for HER2 positive solid tumors, including metastatic colorectal cancer. Again, this is a case of prostate cancer. This is a patient of 61 years old male, and he was detected to have elevated levels of PSA as a part of routine regular checkup, and he underwent prostatectomy, and there is a family history of carcinoma breast at the age of 60 years, and tumor testing was done, which identified a tier 1 alteration in BRCA2. And we know, in BRCA2 variant, so we know that there is an aberration in HRR pathway, and PARP inhibitors could be sensitive for this patient. And here we could see another parameter, which is a VAF, variant allele frequency. So when we see here the VAF of 51%, It is a higher sign and it is indicative of that this could be associated with a germline variant. So where is a kind of a pedometer which will tell you the fraction of how number of reads with the variance by the total number of reads. Again, to confirm that, after pre-test counseling, we recommended NGS germline cancer panel, which detected a germline variant, the same BRCA2 variant, which was pathogenic. And yes, this could optimize patient care to address the hereditary cancer risk and screening for the patient, and also potential genetic risk to family members has been addressed. Next, let's go to a case of chest. This is the case of 52 years old male with a gastrointestinal stromal tumor and went through several lines of therapy, first line therapy with imatinib, then sunitinib, and several surgeries, first diagnosed in 2024, and now in 2025 he started on imatinib again, and he developed multiple metastases and was started in regorafenib. So a molecular testing was done finally, and then uh, it detected mutations in KIT gene. So KIT genes are major oncogenic drivers in known drivers in gastrointestinal stromal tumors. Uh, they instigate activation of c-KIT tyrosine kinase activity and subsequent induction of downstream signaling pathways such as RAS, MAP, PIK3 AKT pathways. Here we could see there's a KIT exon 17 mutation and a KIT exon 11 mutation. So this KIT exon 17 mutation is known to confer resistance to imatinib, which the patient was in. And in the guidelines, it has already been mentioned that nilotinib could be option for better response in exon 17 KIT mutations. And again, yes, KIT exon 11 mutations respond to regorafenib, which also the patient is in currently. Yeah. So next case again of adenocarcinoma colon and poorly differentiated ascending colon adenocarcinoma in a 48 years old female. She was started on adjuvant chemotherapy, and from March 2024, then post-operative changes were noted, and received fifth cycle of Folpox and bevacizumab. Here, the molecular testing revealed BRAF V600E mutation along with BRAF K601N mutation, and which is tier 1. That is, we have FDA-approved therapy guidelines, and for this particular cancer, and tier 2 mutations in ATM genes. The presence of BRAF gene instigate constitutive activation of key downstream pathways promoting carcinogenesis, and encorafenib, which is a BRAF inhibitor, plus cetuximab or panitumab, which is an anti-EGFR TKI. It's guideline approved for colon cancer cases with BRAF V600E mutations. So here we could see that it was approved in 2021 after analysis from the Beacon study. This is the case of lung cancer with this metastatic lung cancer, metastasis to brain, and diagnosed in February 2025. And the tumor tissue was tested, and we detected ALK EML4 fusion. So this fusion is a tier 1 level A genetic variant, and ALK inhibitors, alectinib, brigatinib, ceritinib, crizotinib, confer sensitivity in ALK rearranged tumors. So this fusion also activates ALK and its downstream pathways, and ALK inhibitors control the growth by uh, targeting the L proteins. Here we could see that fusion is detected. And when we talk about detection of fusions, uh, fusion could be detected mostly by RNA or DNA. So RNA-based techniques are quite sensitive and uh, mostly recommended for fusion detection. But yes, if we have a panel which is sensitive for fusions and specifically designed probes to detect that, so DNA could also detect fusions. Yeah. So let's talk about this again. Emerging biomarker in the field of oncology. It's the transcriptome analysis. So we have discussed about mutational signatures. We have discussed about TMB, MSI, HRD, and this transcriptome analysis. How can we look into it for precision decision, right? So by next generation sequencing, RNA sequencing, it enables profiling of gene expression in tumor samples, so we could know if there is a differential gene expression pattern, and that could distinguish tumor biology. For example, in clear cell renal cell carcinoma, it shows high expression of VEGF-CA9 and VHL related genes. It could also detect immune-related signatures and pathway activation that inform therapeutic strategies. So let's look into a case of unknown primary. So this is a case of 62-year-old male with multiple metastatic lesions in the liver, lungs, and retroperitoneal lymph nodes, and the biopsy said it is a poorly differentiated carcinoma from liver lesion, but standard imaging and PET-CT could not define a primary tumor, so it was diagnosed as cancer of unknown primary. And molecular testing results showed that there was expression of the PSA, NKX3, and FOXP13. So these are specific markers that are present in prostate cancer tissue. So it was defined as the diagnosis was defined as metastatic prostate adenocarcinoma. There was also a BRCA2 mutation, so PARP inhibitor could be an option for this patient. Yes, so we've discussed a lot of topics, and with this comes so much data for us. So let's look into now how this data could be interpreted, and how AI can help in this interpretation. Excuse me. Yeah. So why variant interpretation is challenging? So next generation sequencing identifies a huge number of variants per patients, and only a small fraction of it are clinically relevant. Most of it are benign, and some are in the gray area, like the variants of uncertain significance. To manually curate it, it's time consuming and also resource intensive. So we need scalable, reproducible frameworks. So how could AI ML come in? It can scale curation and reduce variability. Is it pathogenic? Is it actionable? Will we report it or not? Yeah. So how the AI models work? So artificial intelligence is a term coined like 70 years back. So its main aim was to create intelligent machines. Intelligence of computer robot to perform tasks as humans, as they are based on human intelligence. Machine learning is a subset of AI which focuses on data and algorithm evaluation as humans do, but in a better way with higher accuracy. Again, the subsets are deep learning, which is a subset of ML. So that allows multiple number learnings from number of data points. sets. So these are complex algorithms. And neural networks are also a method of AI which empowers computers to perform tasks as human brains. So different machine learning models predict different work in a different way, and they predict pathogenicity. So there are different features which are put into the model, which could be sequence context, allele frequency, conservation, functional assays, and the model will give an output whether to report it or not report it. So AI is essentially capturing expert reasoning at a scale. So this is just a simple example where weak AI, like even language learning models like, say, for example, ChatGPT, could assist in a variant reclassification. So we have a VUS, the uh, ChatGPT language learning model will interpret if there are any functional evidences, if from that point of time, from 2020, if there are any functional assays, if there are any family segregations has been done. So it will reproduce the evidences to us, and of course it will support the reclassification. So this is an simplified example, but yes, real world machine learning models work on big data sets and use complex algorithms. Yeah. So this is one of the early studies where AI model has been trained on 19,000 plus variants on decisions made by 6 expert board certified pathologists. So this gives the AI model gives a score 0 to 1 for each variant. Higher score is going to be more likely to be reportable. They use a simple logistic regression model, and very reduced error rates were seen. Again, it was validated on 16,000 plus new variants with strong performance. So here, AI has been used to capture expert decision, and it was reproduced consistently and helped to create a decision support tool. Moving forward, this is a recent study of 2025 where large-scale AI variant reporting was done for 3 oncology assays, and AI trained on over 1.3 million variants from 10,000 plus patients. They use advanced models to predict reportable variants, and the AI learned from trusted databases like OncoKB, Cosmic, Clinver, and this was able to achieve accuracy of up to 99.5%. And one important thing here is that AI is always referred to as a black box. But this study used shapely plots which could say why a variant was flagged, making it easier to trust the AI. So moving forward, explainable AI is what would be helping clinicians to ensure transparency and trust in making clinical decisions. And this is one again of one second. John Hopkins study of dynamic reanalysis and impact on treatment recommendation by AI assisted reanalysis on 2,219 patients, and they retrospectively reanalyzed tumors and data over 3 months, every 3 months and over a period of 9 months. So we can say that tier 1 therapy eligibility FDA approved increased from 11% to 21%, which doubled, mainly because in 2022, due to the approval of TM based tumor agnostic marker. Again, 6% of patients had treatment recommendation change due to updated variant pathogenicity classification. Again, the database is used, yes, AI use continuously updated knowledge bases like OncoKB, Cosmic, Clinver. This study does support that serial reannotation improves strength of therapeutic recommendation and reveals substantial changes in available therapies. Uh, One pitfall could be that there was no clinical judgment by humans. So that is one thing, or we can say data sets could be biased. And when we talk about the promises and pitfalls, yes, AI benefits, genomic medicine and would accelerate variant interpretation with faster, consistent workflows, enabling us to report with a shorter turnaround time. And but again, it comes with challenges of implementation. The bias of data sets in which it has been trained. For example, the black box model of lack of explainability and ethical concerns are also posing because there is risk to equitable and trustworthy AI use in medicine. So moving forward, we can say that AI-powered precision oncology could help in integrating multiomics data, be it DNA, RNA, protein, or and will help assist uh, clinical decision making, but that has to be guided by ethical integrity and sound judgment. Yeah, this is what I have for today. And any questions, I'll be happy to take.
Yeah, thank you. Thank you so much, Nandini, for a wonderful talk. That was an awesome one. Yeah. The room is open for Q&A, please. Simon, would you please take forward? Yeah.
Yeah, definitely, Prashant. Thank you, Nandini. It was an excellent, very extensive and good talk. Thank you.
Thank you so much for having me here.
Yeah, any questions,
Please,
please
the
raise your
platform
hand
is,
and please unmute your microphone and you can ask, please.
yes, please.
Okay,
There
so
is a question
yeah.
for you, Nandini, in the chat. There is
Mm.
Chef and B, and she's saying, sorry, the concerned person is talking about tumor
Mm.
agnostic biomarker slide.
Mm. Mm.
And they wanted to,
The tumor in the slide, is it agnostic? Is it agonist? Oh, it is agnostic. So when we talk, what is agnostic biomarkers? So when we talk about agnostic, the word originated from gnosis, which is it's a Greek word. So Greek word, and that means agnostic choice of therapy means the reliance on the molecular data, irrespective of the knowledge of the cancer tumor histology. So these biomarkers has been approved regardless of the cancer type. But yes, that has to be taken with a pinch of salt because when we say TMB in colorectal cancer, we have seen that for MSI stable cancer, it has not been much responsive. But for MSI, so TMB, so those are tumor agnostic biomarkers. So we have been, it has been approved for all types of cancer, but yes, the clinical decision has to be taken along with other biomarkers also taking into account. I think I have answered. Shafin, yeah.
any other questions, please? So there is a question from Bilal Ahmed. Did you try alpha genome for variant annotation?
Uh, I have not tried variant annotation. So this alpha genome for variant annotation, of course, this the bioinformatics teams mostly use that. So I do not get involved in variant annotation.
So in the
Nandini,
meantime,
good evening.
Nandini,
Hello.
yeah, sorry, Kausi, go ahead. Sorry, yeah.
Yeah, yeah. First of all, thank you so much for your, precisely a wonderful presentation, and with this case presentation as well. There's a very interesting last few slides where you have shown, how AI assists in this variant analysis and all. So, as per your experience so far, how is like accepted to the clinician as well as patients? Like, if you say, for example, lung cancer nowadays coming up in a big way all over the India. So, how they are responding if you're using this AI tool, and if you're not using the AI tool, what is the difference between these two? In the same case, let's say case is same, but at one time you're using the tool, and sometimes most of the time if lab or any diagnostic application, those are not using the AI. So what is the difference in the patient outcome and all that? I just wanted to understand from your perspective.
Okay. So when we are using AI, definitely for the patient, what it matters is clarity, right? So if you are giving a treatment recommendation without having enough evidence or supporting studies, so that could be not useful. So AI would be used to look into the variants, look into several databases, or take help for the clinical judgment in complex cases. But yes, it would not replace. Oh yeah, so it would not replace any. It will not replace clinical judgment. So even if we use AI, but then we always make it explainable to the patients, and
So so
there has
country
to be.
like us, like India, like country like us, like India, still majority of them they're not using AI.
Hmm.
Uh, I mean, I'm just talking about, let's say, any key hospitals, those where patients are diagnosed, they're not, AI is not adopted actually for variant analysis so far, most of the places actually. So probably you will be the, torch bearer for this thing, we have to, you can say, by using this kind of platform, I think we can educate more and more so that this powerful tool can be used and assisted in, this better patient care and all, and better treatment outcome. So this is one
Yes.
thing, and I think Soham is with us. So for all genetic counselors, those who are over here, is there is a Prashant? There is a. I just wanted to announce one this pre-conference workshop that we are going to do in Jabalpur in this Tribal Research Institute from ICMR. So the whole idea is to bring both a geneticist and the clinician on the same platform, and there is a chance to interact one to one also on that forum. It will be in the mid of November this year. So in BICS, most of you know those who are from the Bioinform backgrounds. They are the Asia's largest society, headed by founder member of this Bioclues is Prashant is with us. So this GCCI consortium is came into existence in 2020. I think some of you already know about this. And the whole idea is to bring this technology and bring this resource person also to train the young, geneticist or genetic counselors actually, so that they can bridge the gap between both the patient and as well as the clinicians actually. So Nandini, I think you have a good fan followers also. I can see today also like more than 50 participants were there, and mostly they are from the GC background. So one thing always pitched me actually, Prashant, I always say end of the presentation. If you look at the participants, still northern part is still missing actually. This most of the GCs are from west or south actually. So this is what we are trying our best to have something in this zone as well, so that we can, bring some more and more genetic counselors to this area, and GCCA is going to give. I mean, I think is going to play important role to
Yeah,
bring some more genetic counselors in this region. So Prashant, would you like to add something else
yeah,
that
yeah,
I'm
yeah,
missing?
yeah. On a lighter
Yeah,
note,
yeah.
on a lighter note, there is a GC bias in south and probably in the east. There are not many, GCs, genetic counselors, or GC percentage is missing probably in the north. I'll have a quick point here, Kausak. So the thing here is AI is being interpreted by many, but what I meant was there is a distinct difference between interpretation of AI for variant prioritization or for the for the clinical diagnostic case, in this particular
Yes.
case, the genetic testing. So for genetic testing aided by AI, where many clinicians are already using it for that matter, but I beg to slightly disagree that the large learning models is not the solution for the clinical odyssey here. I don't think that is going to be uh, I think a killer where it might probably give a wrong assignment simply based on you know the chat GPT what it says. But the AI interpretation, which we are also working, and we have also been working with our collaborators in Germany over the last 1.5 years, and even our PhD fellows are also working on the various solutions for this. AI interpretation based on the agnostic models. A, as wonderfully put by Nandini, A means absent, gnostic means the real lack of the knowledge taken from the lack of what do you call unorthodox way, I would say. Okay, that's an agnostic way. So for those particular things, we needed a large amount of true positive variant interpretation, which means we needed those particular variants that are truly called, whether or not they are pathogenic, likely pathogenic, or even various, if not conflicting interpretation. Then a machine learning model can be taken up, and where AI can be predicted. So these days, even Franklin, AI bots, and all those kind of things are already there in use. Many clinicians are using, but they're not really up to the mark. So that was my only point, Nandini.
Yeah. Still not in clinical setup like in hospitals, or because to generalize that also we might have results which are false positive, right? So that's why it's still under research, and of course it will take time to integrate such platforms for all the patients.
Yeah, very much. Yeah, I mean,
I have
yeah. So my
this,
yeah. Sorry, someone is saying, Kausar?
there is a question, I think.
Yeah,
Yeah, Nandini,
Ashna.
there is a
Ashna,
question
please
from
unmute your
Asha.
microphone and ask.
Yeah, and I think there are no other questions other than that. So, Ashna, if you would like to ask yourself, you can unmute, or I'll read out for you.
Yeah. Good evening. That was a wonderful presentation, Nandini. Thank you so much for your time. I just have a one question because you also mentioned about liquid biopsy and the case you presented was very interesting. So, what are your views on using liquid biopsy as one of the earlier testing methods for solid cancers? Usually, what happens for solid cancers, IHC and other tumor testing, gets prioritized. So, do you think it should be used in combined with tumor testing from the starting, or a doctor should wait till the cancer progresses, or after seeing some kind of improvement in the patient, or if there is a poor prognosis involved, then only liquid biopsy should be done?
So that's a great question. But when we see in the recent practices, mostly when we see by clinicians also, they take tumor testing as the first uh, step. The first priority is tissue testing. Again, in some cases where say tissue biopsy is not visible or tissue has failed, or in advanced cancer where we want to see treatment monitoring, for example in breast cancer, say the patient was on CDK6 inhibitors and then the patient is progressing, so we can see if there is any ESR1 mutations like through liquid biopsy to see if it would be resistant to the CDK4/6 inhibitors, right? So these are some of the cases. And again, in lung cancer, again, in NCCN has said that it could be used complementary with solid biopsy. Then even in colorectal cancers to see if uh, after if there is minimal residual disease. So most of the cases we have. When the cancers are advanced stages or we do not have option for tissue biopsy. But yes, if cost is not a matter, availability is there, then both together will give more better results in variant detection. because sometimes some variants might be present in tissue, might not be present in liquid. So doing both will could definitely give a better detection.
So thank you so much.
Thank you, Nandini.
Thank you. Yeah.
I think we are running out of time. So, any other questions, please?
Yeah, so for last, I would like to invite Nandini also to join our GCCI
Okay.
forum actually as a member.
That
So
would be
they,
that would be great. I would
yeah, because
love
that will be that will be a
to.
great value addition to us in the era of precision medicine. So we
And
need
of course, I would be also
trained,
grateful
yes,
that if I am involved and be a part of it.
yeah. So that is one thing, and the last thing, I think, Shwam, are you there?
Yes, sir.
Soham, are you there? Yeah. So
Yes,
would
sir.
you like to say something about this upcoming this pre-conference workshop? I mean, anything that you would like to share because you are the one who is always being proactive for BCGI, pre-conference workshop most of the time that I can see. So for the
Yeah,
first time,
thank
we
you
are going
so
to
much,
do
sir.
it on site actually,
Yes, sir.
and so let's see. We'll try our best to do our best actually in this pre-conference workshop also.
Sir, whatever I have seen till the date about the plannings and the session and the titles, all are very exciting and impressive towards the students. So if we can implement it in a way what we are thinking, so that would be a great impact on the future genetic counselors.
Yeah, thank you, Krishna. Prashant, so we can wind up or
Yeah.
anything else? Saima would like to say something because she is also very proactive when she was in James to me. She was working for the corporate as a counselor also, as a
Yeah.
resource person for the technical level as well. So she can also add something and then we can end the session.
Yeah, maybe some a
If
quick
there is
points
no
based.
further question
We
from
don't
anyone,
have
yes.
time. Unfortunately, we needed to move to another meeting. Sama, please. Yeah. Any quick
Yeah,
points?
no, thank you. Thank you, Kaustav. I really enjoyed your presentation, Nandini. It was very informative. And though I deeply don't go into cancer genetics work, I'm more very, let's say, a germline person in terms of their interpretation and other rare disorders. But yes, the presentation was wonderful and it has given a lot of insight about how the things are going around in terms of AI and everything. Thank you so very much. And I
Thank you
guess
so much.
we're running out of time and there is another meeting ahead. Prashant is eagerly waiting to start. So we would like to have a photograph as per requested by Prashant if everyone
Yeah.
can
Thank you so
switch
much,
on their
Sama. Yeah,
me.
Nandini. We usually take a photograph with your permission. So all others,
Yeah,
kindly switch on
definitely.
your camera to have a good photograph, please,
And
very quickly. Yeah.
thank you once again, Nandini. It was a very valuable presentation. Thank you
Yeah.
so much.
Yeah. Great
Thank you
presentation,
so
Nandini.
much.
Thank you so much. Yeah. Yeah.
Yes. Oh,
Yeah.
sorry. I'm in a very bad
Yeah,
with.
Pooja, Saurabh,
Okay, fine.
Rupesh, Rajeshi, Shweta, Pratiksha, Shruti, Anuj, Shafin, Ashna, please. Yeah. Yeah, thank you so much. And can we all say cheese? जयपुर से लेके ऑस्ट्रेलिया तक दिखाना चाहिए हमारा चेहरा एंड हमारा फेस हमारा नो यस यस थैंक यू सो मच let's give
Thank
a
you
big
so
Thank
thumping
you
round
so
of
much,
applause to
everyone.
Nandini,
Thank you,
please.
everybody.
much, guys.
Yeah, thank you so much. And we have in conversation, whoever wants to attend, please join. So Kausar, he has also left. No, he's
Yeah,
here.
yeah, I'm
Yeah,
here.
he's here. So,
Okay, okay, congratulations. Okay,
uh,
bye, bye.
congratulations. Yeah, sorry, Kausar, I need to leave, in organizing this thing. I'm sorry, we'll probably meet. Sama, are you there? Sama has also left. Sunil, Soham Banerjee, and Kausar. You three can probably discuss. All of this kindly quit. I'm sorry, I need to leave.
Sure.